We are from Group 4. We are going to present the proposal of open-ended lab on the absorption rate of methylene blue dye on activated carbon. For the problem statement, this experiment is carried out to determine the optimal concentration and pH value for the maximum absorption rate of activated carbon. There are two objectives in this experiment. The first one is to study the effect of pH of the adsorbate solution on the absorption rate of the activated carbon, and the next one is to study the effect of adsorbate concentration on the absorption rate of the activated carbon. Next, for the experiment variables, the manipulated variable is the concentration of methylene blue, and the responding variable is the pH of adsorbate concentration, and the absorption rate. The constant variable for this experiment is the contact time.
The following shows the complete experimental procedure of different pH of adsorbate. A 20 ppm methylene blue solution with pH ranges of 1, 4, 6, 8, and 10 was prepared. 0.6 gram of activated carbon was used as an absorbent dosage for the 100 ml solution prepared. Each of the 5 conical flasks with the aforementioned concentration was allowed to have contact time of 50 minutes. Next, the pH was kept constant by adding necessary amount of sodium hydroxide to keep its basic nature and sulfuric acid to keep its acidic nature. Lastly, using a UV spectrophotometer, the percentage of adsorbents at a wavelength of 664 nanometer was determined. The following shows the complete experimental procedure for different adsorbate concentration. Firstly, 5 different concentrations of 100 ml of methylene blue solution were prepared. 0.002 gram of methylene blue powder is dissolved and diluted with distilled water to form 100 ml of 20 ppm methylene blue solution. Step 2 is repeated using 0.004 gram, 0.006 gram, 0.008 gram, and 0.01 gram of methylene blue powder to produce 20 ppm, 40 ppm, 60 ppm, 80 ppm, and 100 ppm of methylene blue solution. Each solution was mixed with 0.6 grams of raw material in separated conical flasks. The samples were then agitated in a shaker for 50 minutes at a controlled temperature of 35 degrees Celsius and a speed of 115 rpm. At regular intervals of 50 minutes, samples were collected from each flask. Lastly, using a UV spectrophotometer, the adsorbents at the wavelength of 670 nanometer was identified.
I would like to talk about the expected results when different pH of adsorbates are used. So in our experiment, the pH of adsorbate solution are 1, 4, 6, 8, and 10. The reason for this is that from the literature, it is evident that the absorption of methylene blue is highest in the natural range. Lower absorption rate of methylene blue at acidic pH can be due to hydrogen ions competing with the cationic charge of dye solution for absorption on active sites of activated carbons. This goes the same for basic medium where hydroxide ions are competing with the anionic charge of dye solution for absorption. The optimum pH is expected to be 5. Explain on the expected results for the concentration of adsorbate. So it is expected that maximum absorption of dye occurs at low adsorbate concentrations, while there will be a gradual decrease in the absorption rate as the adsorbate, which is methylene blue, concentration increases. This is due to the fixed concentration of adsorbent, which is activated carbon, which provides fixed number of active sites. As the saturation of methylene blue adsorbate might be achieved at low concentration, thus increasing concentration of methylene blue will not show in increasing amount of methylene blue being absorbed. Thus, the absorption rate becomes lower. This experiment consists of 5 safety precautions. First is to wear complete PPE such as lab coat, gloves, and safety goggles before conducting the experiment. Next is to handle methylene blue dye and activated carbon in a well-ventilated area to avoid harmful vapors or dust. Carefully label all the solutions and chemicals to prevent confusion. In the event of spilling, clean the area immediately and seek help from the lab assistant. Before placing the cuvette in the UV-vis, clean the clear surface to remove fingerprints or dust to ensure light penetration for an accurate reading. That's all from us. Thank you.